My experiment was done on the neural dynamics of novel scene imagery. Episodic memory formation and retrieval are two of the main functions of the hippocampus. Mental generation of scene imagery is a major component of episodic memory processing. Cognitive impairments that involve recalling past experiences have been proven to increase following damage to the hippocampus. This includes a lack of imagination or futuristic thinking. The hippocampus is also associated with the flexibility of predictive or fictive representation. It has also been suggested that the hippocampus constructs scene imagery in order to facilitate recollected or imagined mental images. Scene in this context is defined as a natural three-dimensional spatially coherent representation of the world, typically populated by objects and viewed from an egocentric perspective. Another region of the brain that is necessary for scene construction is the ventromedial prefrontal cortex, also known as the VMPFC. Image 1 shows the exact location of the hippocampus, and image 2 shows the location of the VMPFC in the brain. This image shows the two proposed models of effective connectivity between the coherent peaks in the hippocampus and the VMPFC. A proposed mechanism of interregional communication is auxiliary coherence. Increased theta sequency between hippocampus and VMPFC has previously been observed during episodic memory retrieval and integration, as well as memory-guided navigation and decision-making. Analogous connectivity during the imagination of novel scenes would provide both evidence that episodic memory and scene construction share similar loci of brain activity, as well as both are supported by comparable network dynamics. Electrophysiological experiments in rodents have suggested that during initial contextual memory formation, hippocampal activation precedes that of VMPFC. By contrast, retrieval, detection of violations in learned information in humans, and subsequent extinction have been characterized by VMPFC driving hippocampus. It is still not completely clear which pattern the generation of novel scene imagery might follow. Although it has been proposed that the mental generation of scene imagery is a crucial component of memory and episodic memory processing, it is also predicted that there is an interaction between the two brain regions during the construction of novel scene imagery. The purpose of this experiment is to find answers to questions such as, first, do the anterior hippocampus and VMPFC display coherent activity during the imagination of novel scenes relative to single objects, as well as, does one of these regions exert a stronger influence over the other during scene imagination? 22 participants, 14 female and 8 male, took part in this experiment, and only native English speakers were recruited. 75 scene words and 75 object words were used as stimuli for the imagination task. In order to enable active imagination, all words were rated as highly imageable. Object imagery was included as a suitable control condition for scene imagery. Additionally, 75 number stimuli were also deployed in a third condition involving counting in order to be used as a low-level baseline. Subjects were told to describe a certain word and create or make up a detailed scene in their mind about the word for 3 seconds. For the object task, the subjects were told to picture the object that was spoken as if it was floating in space for 3 seconds. Participants then heard a beep and opened their eyes. For scene and object trials, they were asked, "What did you imagine?" If they failed to perform the task, they selected unsuccessful. If they did perform it, then they would select either low detail scene, high detail scene, low detail object, or high detailed object. After the scan, participants took a recognition memory test. They were presented with scene and object words and asked if the word was previously presented in the scanner or not. The image below also shows examples of the trials. In order to obtain results, we had to compare the percentage of correctly recognized scenes and object values and analyze them using paired samples t-test. To rule out the possibility that differences between conditions and subsequent analyses were related to differences in eye movements, we computed the variance of these two EOG signals during each trial, which served as an indirect measure of saccadic activity. These variances were averaged across 3 trials within each condition and normalized within subjects so that values for 3 conditions summed to a 1/3 repeated measures ANOVA. To estimate differences in power between experimental conditions and source space, the linearly constrained minimum variance, also known as the LCMV B-former, was used. These images were entered into a second-level random effects 1/3 ANOVA and SPM to investigate power differences across conditions. For each subject, we then performed a linear regression with the hippocampal segment as the predictor variable and the difference in theta power from baseline as the dependent variable. For in-scanner task performance, the participants' self-rated performance was high, with a minimal proportion of trials rated as unsuccessful. There was a similar proportion of scene imagery, object imagery, and counting trials. For post-scan recognition memory tests, the participants were able to correctly recognize most of the scene and object stimuli that they previously imagined in the scanner, performing significantly above chance level in both conditions. They remembered a greater proportion of objects than scenes. For source space power changes during mental imagery, we first determined which brain regions were active during imagination in general. Due to the obvious disparity in task demands between the imagery and baseline conditions, we used a conservative whole brain family-wise error-corrected threshold of p less than 0.001. A widespread change in theta power was observed during mental imagery when compared to the baseline task. This was evident throughout the left anterior temporal lobe, with an activation peak at the whole brain level in the inferior frontal gyrus. In summary, scene and object imagery appeared to engage a common network of brain regions to a similar degree, including the anterior hippocampus. Therefore, any observed differences in connectivity between the two imagery conditions could not be explained by differences in power. Having established the peak locations of power changes in the anterior hippocampus during mental imagery, we then investigated whether the imagination of scenes was associated with greater connectivity with any other regions of the brain when compared to object imagination. We found higher theta coherence in the left fusiform gyrus and parahippocampal cortex during scene imagery. Two of the main statistical tests that we found had important findings. The ANOVA test did not detect any differences in eye movement between conditions with a p-value of 0.335. Using the t-test, on average, we found that subjects remembered a greater proportion of objects than scenes with a p-value of 0.048. Our results also investigated whether the imagination of novel scenes is supported by hippocampal VMPFC dialogue. Mental imagery, either spatially coherent scenes or isolated objects, both resulted in comparable theta power decreases in the left anterior hippocampus. Theta coherence between anterior hippocampus and VMPFC was significantly increased for scene compared to object imagination and was of a higher magnitude for scenes that were subsequently remembered or imagined in greater detail. The observed coherence was also specific to the theta band. The first graph represents the gradient of theta power change along the hippocampal axis during mental imagery. The magnitude of this power change increased significantly from posterior to anterior segments. The second graph displays that coherence between the VMPFC and hippocampus was significantly higher for subsequently remembered than forgotten scenes, with a p-value of 0.018. A similar trend was observed for scenes imagined in high versus low detail, with a p-value of 0.058. These figures both show the dynamic causal modeling of the interaction between the hippocampus and the VMPFC. Figure B shows the results of the Bayesian model comparison, which indicated a stronger influence of the VMPFC on hippocampal activity during scene imagination. Figure C shows log Bayes factors for each participant. The blue bars indicate positive to strong evidence for VMPFC driving the hippocampus, the model which most consistently fit across participants. Green bars represented the only two cases where evidence of the hippocampus driving VMPFC was observed. Gray bars represent the remaining participants where there was no conclusive evidence for either model. In conclusion, this interaction reveals that the VMPFC drives hippocampal activity during the scene construction process. The analyses of event-related spectral perturbations show that the activity of VMPFC precedes that of the hippocampus. The rapid initial engagement of the VMPFC, in contrast to the slower kinetics of hippocampal activation, provides additional evidence that the VMPFC could drive hippocampal activity to facilitate the construction of scene imagery. These findings corroborate fMRI studies demonstrating that the anterior hippocampus contributes to the mental construction of novel scene imagery. This provides further support for a shared dynamic between the two. We discovered that the magnitude of coherence between these two regions during scene imagination predicts whether or not the stimulants would be remembered, while also being unable to suppress context-irrelevant information while confabulating suggests that the VMPFC could also control both hippocampal input and output during scene imagination. Our time-resolved analysis of VMPFC activity during the construction of novel scenes identified additional periods of early and late engagement relative to hippocampal activation, adding credence to this view. We conclude that episodic memory and imagination share fundamental neurodynamics in the process of constructing vivid, spatially coherent, contextually appropriate scene imagery is strongly modulated and moderated by the VMPFC. If this experiment were to be conducted again, it would be beneficial to include more areas of the brain that also involve storing and making memories, such as the amygdala and the cerebellum. It also can be beneficial to include more thorough ways of testing the imaginative scenes, such as having the subject describe them to the experimenter and having the experimenter then rate the scenes. The last critique I had would be to increase the sample size of the study. Since there were only 22 participants, it is hard to make these results applicable, and having more people participate would help achieve that.